So we start with that process in which we think about what is it that we want to answer. How do we design this experiment? What are the best tissues to get? And then, in a very simplified way, you prepare your samples, you prepare your libraries, and you start the sequencing project. As Morgan was saying earlier today, there's always a quality control check at the end of all of these little stations. There will be a process of assembly, which is a little bit what Chris was alluding to in the earlier part of his talk. The automated annotation that Martin was describing, the manual annotation that we're going to do in the next part. And then after that comes, in some cases, not everybody conducts it, but some groups actually conduct a merge so that you can use all of your manual annotation efforts with your automated efforts. And that's what we'll hopefully bring together so we have a consensus set that we can use for our analysis. Some people actually start conducting some of the global analysis before there's even a manual annotation effort. Hopefully we'll actually make things in an organized manner so that we have a solid consensus gene set before we conduct even the global analysis. At the end of the glory, the synthesis, the dissemination, the papers that we give to our community so that we have a better understanding and they have a better understanding. Come on. What is it with you? Okay, well, I guess we're done. Then in order to think and get your mindset into what the process of manualization is, these are some things that I think you should take into consideration. We don't need to review those right now, but these are things that you will be thinking about it. We're going back to basics. I know that this is a crowd that is very biologically knowledgeable, but I know that I sometimes think, what is it? And so let's go back to remembering those things, central biology. Let's think what a gene looks like, and the process of translation and transcription. I am hoping that this computer will behave, and that by the end of this talk, you will walk away with a few concepts and a few ideas solidified. How do we understand the process of genome curation? And how do we go from the assembled genome to the manual annotation model? How do we become familiar with the functionality of web Apollo? What is the best way to approach it? And how do we identify the homologs of the genes of interest that I want to go and annotate? There are two different routes that you can use to get into Apollo with your gene of interest, and we'll learn those. And at the end, we'll try to have a better understanding of how do we look at this gene and do we assess whether we need to correct it or whether it's actually accurate and if it's the best representation of the biology of this gene with that organ. So let's go back to the early days. This is not to insult anybody's intelligence, but actually to bring back a concept that is being evolved for 150 years. And it's a very malleable concept that you study every time you teach or that you study when you sit down for your classes. And throughout the talk, I'll probably go several times and ask a question such as, have you ever heard of the sequence ontology? And I would like for people who haven't heard of the sequence ontology to raise their hand. Because I want to see what's the, 1, 2, 3, very good. And so in that way, I'm getting a gauge of how much am I talking to the crowd, things that everybody knows, and how much I am kind of diverting from the expertise. So like many other things that we have come across, data needs to be standardized. And the sequence ontology is a form of standardization of the elements of the genome in a controlled vocabulary manner. Basically, it says that what you understand as a gene is what I will understand as a gene. And it's only computationally. It doesn't mean that it's all the truth, but it's what we can actually reduce into a box, and so a concept that we can all identify and understand. So a gene is then defined by the sequence ontology as a locatable region of genomic sequence that corresponds to a unit of inheritance. And it's associated with regulatory regions, transcribed regions, and or other functional sequence regions. If you're more interested, especially the students in understanding more about how this concept evolves, there's a very cool graph in there from genome biology. In our lifetimes, actually, this concept has changed drastically thanks to the efforts of many projects, especially the Encyclopedia of DNA. And they yet again shifted the way in which we understand the gene. And so now we have basically the understanding of a segment that contributes phenotype and function, and in the absence of demonstrated function, and it's very important, a gene can be characterized by using sequence, transcription, or homology. So we are actually going to use sequence similarity approaches in order to be able to reach into the genome and grab the homolog of the gene that we're interested in. We don't all have the ability to go in the lab and conduct experimental efforts to figure out what the function is, but we're going to make assessments based on sequence similarity. So consider these three things. A genomic sequence directly encoding a functional burden. This is very important. If several products share overlapping regions, it will take a union of those overlapping sequences, those overlapping genomic sequences, and the union must be coherent, that is processed separately for final protein and RNA products. But it doesn't require that all the products necessarily share a common subsequence. Okay? That sounds very abstract, so what does that look like when we're looking at the genome? At the top, we have the DNA sitting on the top of the genome. On your chromosome, we have the two strands. And on the forward strand, in this case, 5'3" on the top, we have several primary transcripts that are being produced. So we have this transcript and that, and then we have this lower one. When they are spliced, after the transcript is actually processed, we have this set of two transcripts over here. These transcripts are sharing some coding sequence in here, and these transcripts are sharing some coding sequence in here, and these share some coding sequence in here. Eventually, the protein products show us that those are all isoforms, spliced variants of the same gene. On the other hand, we have two of those spliced transcripts that share the unrelated regions but do not share coding sequence. So even though they have coordinates that overlap with each other, we still consider those as two different genes. They will be two different proteins. And finally, we have non-coding RNAs at the bottom. So there is no product being produced in here. It is also considered yet another gene. So think about that when we're looking at the mRNAs and we're actually trying to figure out what's the best representation of my gene of interest. Another thing that we'll need to remember is this concept of open green frames. Earlier, Chris was talking about it. Who remembers open green frames? Okay, we still have some that are fishy about it. So what's important to know, you'll have this entire slide for you, so you can read the rest of the stuff, but what I really would like to say right now is that in eukaryotes, only one open green frame is biologically important at a time for the DNA. So you're only going to be inspecting one open green frame. The software that goes and tries to capture what those genes are will actually attempt to figure out which one is the longest one within the structure of that gene. That'll be important because every time you conduct an edit using software for editing your gene annotations, the software will actually recalculate that open green frame every time. So after every edit, you'll have to check whether or not the frame has been changed or subcodons have been introduced. Then another thing that I'd like to remind you just now, again, this is more information about translation, but the one most important part that I would like to remind you of is splicing signals. If you're an intern and you're sitting down in the middle of two exons, to your 5' end, there's going to be a signal that is usually canonical, it's a GT. And then to your 3' end, when the other exon begins, there's going to be an AG. Why is that important? Because you are going to be reviewing the boundaries, internexon boundaries of your gene model so that you're able to figure out whether or not they are canonical and they are respecting what you know and what you know about the biology of the organism. So that's a canonical splice, GTAG, from the point of view of an intern. Hopefully that paints this picture in your mind now. There you have the exon, and remember that the untranslated regions are actually part of the exon. You have some introns that are later spliced, and then at the 3' end on the C terminus, you have another exon with some untranslated regions. As we heard earlier today, not all of the prediction software actually managed to give you information about UTR, so we have to go in and sometimes manually add them. And the last concept that I want to bring back to your memory so that you can remember this is phases and frames. So the intern actually has the ability to interrupt the reading frame at different portions of the codon. It can go in between two codons, or it can go in between the first and second nucleotide, or between the second and third nucleotide. And depending on that, there represents 3 different phases. This will be important not because of the phases, but because of the reading frame when you're going from one exon to the next. And you'll have to remember that because when you're looking at the exon boundary, you see you will see the actual amino acid sequence on top of the nucleotide sequence, and then when you go to the other exon, you'll be able to make connections on whether or not it is correctly reading in the frame. Usually the gene prediction gets that very well, because we actually have good models that allow us to figure out how to splice the gene model within. Is that portion of the structure of does this really belong to the gene or not, that gets a little fishy, but this part is usually correct. However, when you conduct edits, sometimes there are going to be warnings that tell you that that the gene prediction has been altered and then the splice site is no longer canonical and you have to fix it. So you have to remember how to obtain a canonical splice site again. So this is just another pass of then how is it that it works to curate genes. We predicted the gene model, we've done the annotation, so the putative assignments of function to those predicted models, and now we go in to actually inspect very in depth. That process of automated annotation, as Morgan and Chris were saying earlier, is not perfect. As Wes was clarifying, there are many possible things that are introducing the errors, and the errors would be just simply assembly errors. There's just not enough information. You're going to find gaps. There could be indels that are introduced because of the nature of the sequencing technology that you're using, and lastly, the lower coverage of some of the sequencing will create some errors. And so because that is not a perfect process, then we decided to talk about manual annotation. And so basically what I want you to think when we're talking about the process of manual annotation is that in order to be able to identify with precision and clarity those elements of the genome, we're going to need to go and review a little bit more closely. And we're going to combine those automated predictions with the experimental evidence that has been made available to us to produce very informed gene models. There are two objectives to manual annotation. Be able to identify those elements that best represent the biology of the gene that you're interested in, and also the ability to be able to eliminate as many errors as possible from those gene models. And then the second objective is to actually attempt to assign putative functions through comparative analysis. So again, we're not actually going in the lab and conducting the experiment to get the functional data, but we can actually do some of those analyses by putting a phylogenetic framework around the sequence similarity structures using Markov models, using databases that actually build the tree so that we can compare sequences against them. So this is just one of the examples of how that can be done, so that you assign gene ontology terms to your genes. I am familiar with the gene ontology. Okay, good. So you have heard of it. But this process of manual annotation, as you could probably figure out by now, is not scalable, right? You're not going to sit down and conduct 10,000 manual annotations all by yourself, which is why we're here together. Hopefully we'll attain a list and we'll go through those genes of interest. And so we were reading actually the list of what were some of the interesting genes that you had identified that you would like to hear or to get the community excited about. So all the steroid hormones, I'm just mentioning some of the ones that we got from this survey: plumage, melanin and other color pathways, feather development, pigment transport, elevation gradient, hemoglobin oxidative phosphorylation, neurotransmitters, sex determination, and also cognition. So there's a number of genes and gene families that we're interested in. And so what we will eventually do is to rally together around those gene families so that we can track the process. It used to be the case that you would sit together with very highly trained biologists and very highly trained bioinformaticians, lock them in a room, don't let them out for a week, feed them something, let them sleep, and then you have the Drosophila genes. And in some other cases, you would have a very highly trained group of scientists. About 150 people are the people who form the Genome Consortium. But there's only about 20 of us that actually sit down and manually conduct the annotations and make assignments from the different models. That's a very specific, much like it happens in a museum when the curator of specific family is only in charge of that one. And then the other model of this manual annotation is when you just sit down in your house and conduct annotations, never show them to anybody. So hopefully, you overcome all of that by realizing that this process of genome annotation, as Chris was saying earlier, is actually an inherently collaborative task. And of course, I'm biased about insects, so this is my best example of sociality. And hopefully, knowing that we turn to each other, and that's the reason why we're all together here, to be able to find better insight in some of the aspects that we're not so experts about, will give you a better idea of how to reach into each other's expertise as well to be able to conduct manual annotation. To facilitate that process, then that's where I come in. My laboratory has been developing for 13 years Genome Annotation editor that is now fully based on the web. So we don't have to download anything when we're conducting our annotations. We just go to a URL on the internet in a browser, not use Internet Explorer, and we will be able to conduct the annotations. So it's web-based, and it's actually uses a Genome browser different than the ones we were looking at this morning. Instead of the UCSC browser, we're using JBrowser. It is for those of you that have a Seki gene. It's JavaScript based, and it's very slick. And it automatically generates data that you can just export the data and extract it as it comes out of a photo. It allows you to conduct annotations not only of protein coding genes, but also non-protein coding genes and other elements. And it has a series of gestures and pull-down menus that I'll show you in a little bit so that you can become familiar with it. For those of you even more technically inclined, we've just reformed the entire architecture of a photo. In the back, we now have a faster architecture. We have the ability to actually store our data and to easily query it in a more efficient way. What all of that means for the general public is we can have more than one manuscript gene in a single instance of a portal, which is a really good thing. We don't have to look out and then look back in. If we're conducting more than one annotation, if we're associated with 2 or more projects, then you can run all of those from the same instance. That's basically what that means to us. What we have been doing is training and supporting communities like this one so that we can conduct a collaborative effort. And so I serve usually as a gatekeeper of the annotations that are coming in. And I also do trainings like this one. What we've learned from going through that process is that a little bit of training goes a long way. Like Chris was saying, every single one of us can become a very accurate annotator. It requires biological knowledge, check. It requires attention to detail, which I'm quite sure you all have. And the discipline to actually want to get more information about your favorite gene. So those are positive things that we all find to jump like that and be successful. But we have to enforce strict rules. So when we start conducting this effort, we'll actually have a little bit of an SOP and guidelines on how are we going to name our genes, which are the families that we're going to start with, and how much information do we want to receive in a few minimal points that you will have to check in order to consider a gene finalized. There are additional challenges that we're overcoming as more technology becomes available. Hopefully at some point I'll be able to get rid of that last bullet. So let's dig into what a photo looks like. This is all information that's available on the web. Currently, this is where the user guide lives. This presentation is even more updated than the user guide because we actually started the release on Friday. We're releasing officially the new version tomorrow. And so this is the most up-to-date version of that guide. But this is the general process of curation. These are the 6 steps that I think you take when you're conducting this. You go and find the region of interest. You have your gene of interest in mind, or you don't have a gene of interest in mind. You just go on an expedition and you find the region that you consider of interest, and you check it out and you put it on your browser. And then you look for the amount of evidence tracks that we have in order to support those gene models. What do we have available? Do we have RNA data? Do we have alignments, blast alignments, or do we have other types of alignments with sequences from other organisms? And then one of those tracks will actually be the starting point for us to conduct our annotation. I'll show you in a second what the window looks like. But when I say there's a user-created annotation area, that is where you actually conduct your edits. You'll have a static form of the browser where you will only be able to see the annotations, and then a more dynamic one that you interact with, and that's where you conduct the edits. You'll adjust the gene models if it's necessary. They're not all going to be perfect. And then you'll check that those edits are actually giving you a wholesome protein, and that by that, checking for integrity and accuracy, I mean, have you recovered everything that we know about this protein? And the best way to do that is to interrogate public databases in order to be able to know whether or not you recovered that structure. And this is what the annotation window looks like in a photo right now. This is the most up-to-date version. When we conduct our exercises today, you'll be handling this part. This panel will not be available now for the exercises, but I'm going to explain it because when we load the Manakin genome on Web Follow, this is what it's going to look like. So you will need to understand that panel as well. Hopefully this will give you an easy tour. These are the user-created annotations that I just told you, so this is where you're going to be editing. And these are the evidence tracks. So all of the information that we have available for our genome is going to be over here on the right. You click and unclick or check and uncheck in order to make them appear over here. What we have here on the right is actually a few transcription data displayed as either the reads that are aligned to the genome or as coverage plots or plots, depending on the representation for each one of the regions of the genome. And these, in this case, just represent different stage and cell types. This is from the honeybee. Some of them come from nurse bees, others from forager bees. So those type of different data that you can have. But you can also have, in this case, protein coding genes that were predicted by NCBI in the Genome pipeline. Does everybody know what NCBI is? Anyone who doesn't know, the National Center for Bioinformatics here in the United States, part of the National Institutes of Health. It's a very, very wealthy resource that we'll be tapping into several times during our exercises. And there are many things that are available from there. We have the PubMed libraries where we can get a lot of publications from the biomedical sciences, but we also have the tools to run Smith Waterman algorithms so that we can conduct sequence similarity searches. Basically, I have a sequence, and then I go to that public database and query the database with my little string in order to be able to find similar sequences that come from closely related. When you have your follow window, you're actually able to upload different files. For example, you went back in the lab and Barney had a few more transcriptomic data that he wanted to use that has not gone out to the rest of the community, but actually have been aligned to the assembly. So you can go through your computer and upload it so you can see it on your local machine, even though we're using the internet. These are data that perhaps it's just a little bit of data that you know it's aligned to this particular region of interest and is of no use to send it out, or you want to keep private before you actually release it to the community. So you can do stuff like that. It also allows you to cheat. So when you are trying to annotate a gene model, and there's absolutely no gene models predicted, like in this area or in this little bit, if there are no gene models predicted, you can actually create a file to allow you to make a fake gene model that you know exists because you've gone in the lab and conducted some experimental data. So just keep in mind in the back of your head that cheating little thing so that we can talk later and perhaps walk you through that process if it's necessary. You have the ability to, as you see here, this area of the evidence can be either dark, like you see here, or it can be white, and so you can change it. And the gene models can be all in the same color, or you can color the exons according to the coding sequence. And so each coding sequence will have a different color. With my deepest apologies to those of you who are colorblind, it's still green and red. I'm trying to change that, but there's also blue. So the 3 different frames will be colored green, red, blue, or purple, and they will be colored in both directions. And you can query the genome using BLAST. We sacrifice a little bit of the sensitivity for speed, and that means you are going to have to use longer strings of amino acids when you're querying the genome. So that in these strings of amino acids, we'll have to have closer similarity. We'll have to be a lot closer to the genome in order to be able to retrieve it.
Do you tweak the black and white parameters, or is it just like default parameters?
These are default parameters, but we have an API and you can get into a follow whichever way you want. And so people have customized ways of getting BLAST into a follow instead of using BLAST. And so, for example, I was telling Paul this, that my colleagues at the National Agricultural Library at the United States Department of Agriculture have actually made up a whole interface apart from a photo. That's called, it's a regular BLAST that you know, but it has a little overly steroidal thing on the side called Django BLAST. And so you get your Gecko Blast into a through the web services, and you can use this, tweak the parameters there however you want. But not Blast. Yes.
You say it requires longer sequence. How much longer?
We're talking about for amino acid string, we are going to need 30 residues, but they will need to be a lot closer to the actual sequences. So they cannot be very distant sequences because Black is a little picky about the similarity. You need at least, I believe it's 85% sequence similarity for amino acids, and I can't remember that for nucleotides.
You can tweak Black parameters also.
Yes, you can, not enough of them. Yeah. Does that answer? Yeah. You have, just like you have in any other type of navigator, you have some tools to help you get oriented. You can zoom in and zoom out. You also have the coordinates, so this will become important so that when you're looking at a region and you go for lunch and you come back and if you close your router, you can just go back to the same place, although Apollo will remember the last place that you were in. And you can also use these arrows to move, but you can also use the arrows on your keyboard or use drag. Which is the biggest part because you don't have to wait for reloading. Apollo actually constantly, dynamically loads all of the views. I'll go faster in this. You can search for a gene or a scaffold. I'll show you that we index the names of all of the genes, so you can just type it in that box. And you can also rubber band the region. So if you just want to, if you want to zoom into this region, instead of trying to zoom with those or the right click, you can just drag over this gray bar. Go like that with a rubber band, and then it'll zoom in. And lastly, this is the annotator panel, is the latest part, and I'll explain that still in this slide. And you can also log in and out, which will be important for you to just if you're sharing a machine to get out of your work. So in that annotator panel on the left, on the far left of the you're right, but the far left of the track, you have different tabs that will get you information about what's going on with your genome process or the annotation process. So you have a list of all the annotations that are available. You have a list of all the tracks of evidence that are available to the researchers. You have a list of all the reference sequences, and in this case, reference sequences are all the different scaffolds. So depending on how fragmented your genome is, you can have 5,000 Gloria scaffolds or 150,000 terribly sad scaffolds. And depending on that, you have a list of all of those available. You can also search for the scaffolds instead of having to browse all 150,000 of them. You also can change organisms. And in the case of the instance that we're using, I believe that we have honeybee, but we also have cow and other organisms that you can look into. If you're an administrator, for those of you that will be doing that task, then you can actually change what the users can do, permissions and those things, create groups, and do all other administrative things like get statistics and then reports of everything that's happening with a form. You have a little bit more of information that you will need is in that annotations tab, each one of the gene models, so this is the gene and this is the transcript, right? And then you'll have some information about each of those. This information is automatically generated only for the ID that is generated when you drag it into the user-created annotation, but you have the ability to change that information, and I'll show you in a little bit. And it'll be important because you actually want to collect as much information about your gene as you can. And then you also have the ability to change the boundary of the exons. You can do that directly in the panel, or you can do it from these tabs. You can export all of the information that you're working with. So the GFF3s, I know what GFF3s are. So it's a generic format in which we actually spit out genomic information. So the genome browsers take all of that sequencing assembly in the back, and it takes all of the information about the genes. And the way in which we register how genes are located on top of the assembly is with this generic format. It's a tab delimited format, incredibly inconsistent across all of the sciences, and it's a tab of 9 columns that capture the most information we can possibly. And so we call it the GMF version 3. I can talk about that forever. And then you can export that information so that you can share with collaborators that will be able to look at it in a different screen, or because you're going to conduct analysis with that. and you can also just export the FASTA files. When I say FASTA files, I refer to the actual strings of amino acids or nucleotides, and then you'll be able to just look at those to conduct global analyses or to share them with collaborators that are not able to look at it alone. This is what the screen will look like for you today when we're working with the exercises. This is the first screen. It's called the sequence selection screen. And over here in this box, you can enter the name of the scaffold or the group that you're working with, if you know it. You can organize it by name or by length. And on this end, you have a tab called Tools, which will allow you to search for the particular sequence of interest against the gene. So you query the genome using the search function from that Tools tab. This is one of the examples of a genome that has 35,000 scaffolds. Is not that bad. Okay, so here we go with the functionality. So now that we know what the window looks like, these are the things that we're capable of doing when we're editing a gene model. In order for you to actually get a gene model from the evidence track into the user create annotation area, you either double click on it and drag it, or you use the right click menu. You right click on this information, and then you have this right click menu that tells you all the different kinds of features that you can annotate. From genes to transposable elements and everything in between, we have 9 different kinds of features that you can annotate using Apollo. And then there's this entire right click menu that is available for each one of those annotations. So that menu, is produced in this area only. This is the feature, and then you right click, and these are all the options. So you can retrieve the sequence. During the exercises, I'm going to ask you to get the sequence, and you can get protein sequences, CDS, cDNA, genomic sequence with plus and minus 500 nucleotides on each side, depending on what you're doing. For the most part, you're going to be retrieving amino acid sequences. So remember, that's where I say, when I say get sequence, right click on the you can get the GFF3 file. You can zoom to base level, and I'll show you that in a little video. And you can also pull the information editor, which will be going into detail in a few slides. These are all the other functions that you can conduct with your gene model. You can delete an exon or an entire model. You can merge 2 different exons from 2 different gene models, or 2 exons within the same gene model. You can split 2 gene models or 2 exons, duplicate it, and you can also use the function to set the translation start. This is important. You're going to be looking at your gene model, and sometimes the predictions actually missed altogether where the time. So in order to figure it out, in order to make it static so that when you conduct edits, it doesn't go away, you set it with this function. It's called the translation start. And you can also ask Apollo to statically remain on the same open reading frame and not change it constantly, so that every time you conduct an edit, it doesn't change. Sometimes you need that. Also, if you're working with some cysteine protein, containing protein, then you can actually just tell Apollo, all right, skip through that stuff, make a read through here because this is cysteine, or in any other case in which you would need to annotate with that. There are other functionalities that you will have here. I'll show you these in a video in a little bit. It's just how you use the evidence to move the boundaries of your exon. And then at the bottom, the ability to change automatically the non-canonical splice sites. Remember what I was telling you earlier, you have a GTAG. In the past, you would actually have to go and figure out where the next one was. Now we've made it automatically so that you don't have to try and do that on your own. Thank goodness there's an undo button for everything that we do and we don't like. And if you change your mind, you can just redo it. There's also a history. And so you actually have a list of all of the functions that you performed at each one of your genes. And you can see, if you click on an earlier version of the gene model, it displays down here in your screen. If you click on the lower one, a later version of it is displayed down here. I have yet to upload this, as you can tell. There is now a little box over here that actually allows you to revert to that particular version. If you are now sure that everything you did was wrong, just revert to the beginning or delete it altogether. But it's good to keep track of that and also keep track of who conducted those annotations in case you've been collaborating with somebody and you were trying to figure out what Chris was doing with that gene model, and if I think he did a better job, I just get out myself out of there. Then this is the information editor. So what I was saying earlier, from the right click, there's going to be metadata that we need to collect. Just like we collect metadata whenever we go and capture a specimen, this is how you collect the metadata for your gene. There will be information about the name and the symbol. There will be information about the description. What is it that we think this gene is involved in? When it was created, those things will be captured automatically. And then at the bottom, we have the cross-references. So it is possible that you have some information about the gene in your own database, or that you know that Morgan has a database that has been successfully run for a long time at his institution, and you want to link to an entry in that database, or at least keep a record that this gene is also stored there, so you will use that, and that's a PDX gene. It's just a cross-reference to a database somewhere else. There's also the ability to add gene ontology terms as appropriate. If you've gone and read and curated the literature, and you've actually used the resources that others have conducted and made available to you in order to associate a gene model with a term, then you can use it by typing the standard gene ontology term number in here. And so we're actually connected to the gene ontology database, and it'll talk to it to try to get you a little automatic fill-up menu. And it would also allow you to write comments. This is very important. If you're conducting an annotation and you know that two genes need to be brought together, then you want to write about that so that when whoever's in charge of curating at the end, when we're producing the official gene set, then we'll have information about what happened to that gene model. If we have a gatekeeper, which I imagine we will, then that person in charge of reviewing everybody else's work in a bird's view type of approach will be able to look through this information. So the more thorough you can be about the process that took place to change that model, the better. These are just some of canned comments that I created. They're customizable, and we can figure out what are the best options for our community. When you right-click, you can tell Apollo to zoom into base level. And this is what that looks like. In the middle, we have the reference sequence. We call it the DNA track. Above and below, we have all 6 translation frames. In the upper part, we have the forward strand. In the bottom part, we have the reverse strand. And thus, these are the translation frames that are coming from the 5' to 3' from right to left. We also have the ability to change the color of the coding sequence, as I told you earlier. From the view menu, you can actually give the 1, 2, and 3 frames on the forward and reverse strand a different color. And you can actually toggle the reference sequence. If you're only working with the top part, with the forward sequence, then you can get rid of the bottom one, so it facilitates visualization. You can actually zoom in and out by holding the shift key and just the arrows if you don't want to keep going on the right hand, the right clicking, or with the little magnifying glass.
Question.
Yeah.
So now PacBio data, there's going to be a lot of inbounds. And so there could be frame shift mutations. So can we collect those?
Yep.
Okay.
Yeah. And so I'll tell you a very specific example of that in a few seconds. And I'll save the warning for there. Yeah, we can do that. So I consider that there are some simpler cases and more complex ones, because this is how we annotate simple cases. This is when the predicted gene model is correct or is nearly correct, and you have evidence in support of this, and you're convinced that the evidence is sufficient and tells you that the gene model is the best representation. As we were saying earlier, in many cases, all the evidence that extends beyond the predicted model is actually assumed to be non-predictive, but we will use that for UTR annotation if that's what we're deciding to do. In some cases, it's easier to use it, and in other cases, you will realize that there's just simply not enough reads to be able to annotate that. So these are some modifications that you can conduct. How do you add an exon? So you have 2 ways to add the exon. You can actually just drag it up, and once you drag it up, you kind of bring this ghost towards the gene model where you want to add it. And it'll turn green to tell you that it's ready to receive the exon. If you're adding an exon from the reverse strand to a forward transcribed gene, then Apollo will warn you that you're not doing something biologically correct. And then a confirmation box will tell you that you've done it. And then every time you conduct an edit, you have to check for the start and stops of your gene model to figure whether or not a truncation of the protein may have occurred. To add the UTRs, you can either do it by modifying the boundary of the gene of the exon, and to do that, kind of hover over the gene over the exon, and this black arrow will show up, and then you can just drag it, much like you drag any other element on the graph. The black arrow, you drag it back and forward in order to change what that exon is going to look like. The other way of doing that is using reads from the evidence tracks and just telling the follower to find that read and to make the edges of those two elements match. And that sounds a little vague still, but I will show you in a second and you will understand better. So make sure that you check for exon integrity again and again, and you'll see this coming up again every time that you conduct the edit. So you'll zoom in to the base level and you'll figure out whether there is a, until you can see distinct rectangles for each one of the exons. You can actually click on the feature and use the square brackets in order to advance to the next one, or use curly brackets in order to advance to the next transcript, not the next exon. And then as you go from exon to exon, you will actually look and figure out whether the boundaries are still correct and canonical. In order to bring the edge of one of the exon that you want to modify to what the evidence is saying, these down here are RNA-seq in this case, and over here is your gene that you're editing. And you have now determined, because of all of the reads that are available in the area, that this gene model should actually extend until the edge of the last of the RNA-seq reads. And so what you do is you shift-click to select both the exon and the RNA-seq read, and then you say set as a 5' end. And when that happens, you see that it has changed. You can also see, perhaps not very easily from the back, that there are some red lines at the edge of this RNA-seq read. Perhaps it's better for you in the back to see them over there. And there's a red square that has appeared around the exon that I have selected. There's a functionality called edge matching. And it's going to be very important for you to look at it because it basically looks at the gene model after the user created a specific area, and it browses all of the evidence down below, trying to figure out whether the boundaries match. And if they do, it'll color them red. That'll give you some idea on whether or not the evidence that you're looking at is supporting the gene model that you have, and it allows you to figure out whether it has actually matched the gene model that you just modified. There are several warnings that appear. Apollo has a biological brain that we call the editing logic, and it actually is the part that sets the longest open reading frame for the coding sequences every time. But it also lets you know of this non-canonical splicing, and it colors them as orange circles with an exclamation point in the middle. Again, the edge matching function that I was mentioning draws this red line. Sorry, you can probably see that better in the back. Draws this red line to let you know that the boundaries match. That way you don't have to go all the way to the sequence level in order to be able to see those. And then you just, as I said, drag left or right click to solve those canonical splices. Kind of a repetition of what was happening. You can also set the start and stop, as I showed you earlier. So if I know that this is the first methionine and it is the initiation of the protein, then you can select the A and then say set translation start, and Apollo will automatically remember now that this is the beginning of your protein and that you keep it. In very rare cases, the start is actually not an ATG. And so we don't have a way of saying that a start is not an ATG other than you set the translation start and make a note. So we don't have a flag or a different color for this. So until we do, make sure that if you encounter that, if the gene that you're working with starts with a non-canonical ATG, then you have to report that. If you're actually looking at your gene model and there are no starts identified, start looking upstream, more to the 5' end of the gene model. Sometimes, unfortunately, gaps in the sequences or some windows actually create the hiccup and you're not able to annotate it. But make sure that you take note about that event as well. Okay, ready for some complex cases? Yeah. Okay, we're almost there. Let's I'm keeping track of that. So more complex cases just simply means, well, it ain't that good. So the automated annotation process was not successful in identifying my favorite gene, and what I'm going to do is to bring two gene models together, for example. And to do that, very simple, you click on both of those genes, click right, and and then merge them. Or I can split a gene, the same thing. I know that this gene model is way too long because I have actually analyzed the entire sequence, and I have recognized that there are domains that don't usually come together. And if I am able to recognize that, then likely those two genes don't need to be together, and I split them apart using, again, the right-click technique. Every time you conduct an annotation, what are you going to do? Every time you conduct an edit, you check. You check the status, integrity of, and accuracy of the model, start and stop, always, because the org is going to be recalculated. So here we come to what Jason was asking about. So we are, there are some cases where frame shifts have occurred, we have single base errors, and we need to actually tell Apollo to read the gene without that error, and we modify. What we do is we create what we call genomic alterations or sequence alterations. Now, this is very important that you remember, because I know in the back you're thinking, you're changing the effect, but we're not. Right? Every time that you look at a folder, you're looking at a frozen picture of the effect. And you're not actually able to manipulate it and change it. What you're going to do, much like you do when you go on Facebook and the square appears around somebody's face, you are anchoring metadata into a piece of information of a photograph. So this is what you do with the genome as well. You're annotating metadata into the sequence. However, Apollo is smart enough to actually read that metadata and modify the way in which the sequence is produced. And so if you identify that you need to, for example, add a nucleotide to a particular region, so that in this region I know that I should add another nucleotide in order for the sequence to shift the frame, then I will write down here on the where it says add insertion, create genomic insertion, and then here I add my letter, and Apollo will show me a modified panel of the sequence in which the insertion will be visualized. If it's a deletion, it'll be a rectangle in red that is circling the base pair that is going to be eliminated. And if it is a substitution, it'll be yellow. Either of those, any of those three cases should happen. And what it'll do is that it'll come reading, find the alteration, go and take care of the alteration and incorporate it in the logic, modifying your sequence. So you can very easily just look at the entire sequence before you conduct the alteration and look at the entire sequence afterwards. You're actually going to do that on exercise 7. Does that answer your question? Yes. Okay. There's also the ability to annotate selenocysteine containing proteins. For those of you that may not remember, there is the 21st amino acid that actually is included in some of the proteins. Selenocysteines are encoded by the regular UGA or TGA stop code. And so in order to be able to annotate a selenocysteine containing protein, you just tell Apollo, you know what, I actually know that this protein should continue, and instead of stop, you just right click and tell it to do read through. And so you just set a read through stop, and Apollo will automatically change it. And it'll look like this little purple line. I know it's purple and blue, sorry. In the back, you could perhaps see this little tiny line that the yellow arrow is pointing to. But this is what it looks like, actually, when you zoom to the base level. And that dark purple and the light purple is not very easy to identify. But what happens to your sequence is that it has been extended, and instead of there being a stop, now you have the U that is the representation of this element. Okay, so you can do that as well. I am not going through this slide. All it contains is everything that I just said over the last 10 slides or so. So you have them in there, and instead of like having to go through the video, you read through all of this whenever you're playing with your gene of interest. But this is just teaching that I did. So now we complete this annotation. I've gone in, I figure out what the modifications needed to be. I know that I have to bring some things together or split other genes, modify boundaries. I've added information about genotype terms that are associated with this particular gene. And I know that there are certain entries from Chris's fantastic bird database, and I have entered those information as well. And then I've actually retrieved the sequence, and I have looked at the finalized product and queried the public databases in order to be able to figure out whether or not I've accurately recovered the entire gene protein. Sounds like a lot, right? There's not a whole lot in there. So I created a checklist for you to go through that process. So this checklist allows you to go through the steps of accuracy and integrity, whether or not single slide so that you remember. So if you remember one thing from this talk, print this slide and then you have it next to you. That will you probably use every time. Okay, so let's be ready for an example. Who knows what Hyalela-Steca is? Have you ever heard of this? The microcrustacean, a little amplified crustacean that the EPA has used for a long time as a bioindicator. And so it's a little crustacean that lives under what the level of toxins are in some systems. And so my example is coming from the arthropod world. Unfortunately, we don't have the Manakin genome app, and so I can't run many of the examples, but perhaps you've heard of a voltage-gated sodium channel before, and so that's the example that I'm going to run. So, as I said, I'm going to use this little guy. It's called Hyalela-Steca, and that's what it looks like as part of the initiative to sequence 5,000 arthropods. And it is of no importance for you to see what's in here, but in this slide, I'll go into this in a second. In this slide, I have the URL for the demo. So if you got absolutely no interest in listening to what I'm doing, but later on you kind of feel like, hey, let's go figure out how this thing works, you can actually go to this URL and you go all by yourself and play the entire honeybee genome, and we actually are putting, I think the line title genome is up. We have a interesting looking version as well, and other data that you can play with. That's the one idea. So how do you go about annotating a genome that you've never visited, or if you just play, right? Because you just want to make sure you don't have a specific area of interest, and you're one of the students that you're actually interested in lending some expertise. So you can start by figuring out what do we know about the genome. In the case of this genome, NCBI has nothing. There's, I think, 50 ESPs, and that's about it. And all of the nucleotide sequences for the genome are actually mitochondrial sequences. So there's not a whole lot in NCBI. But we have a wealth of information over in our own system, which is going to be made available to NCBI when we're done. This I5K workspace at NAL, which sounds very acronym-like and not very clear, it's basically the system that we have developed at the National Agricultural Library of the United States Department of Agriculture, where we host all the orphan genomes from the arthropod genomics efforts that don't have a house anywhere. And so in there we have about 10,000 samples with 13,000 proteins or so and 23,000 transcripts for those places. I wanted to know something about this genome because I don't work with it, and I wanted to give an example. So I went to PubMed, and this is a very easy way for you to find a genome interest if you have none. Go to figure out what's the latest in the literature. And so when I looked at the biomedical literature at the NCBI site, I found this paper that actually talks about how some populations of the Hyalela-Steca have 550-fold change in sensitivity to this particular pyrethroid. And so this is an insecticide that is only used in terrestrial systems. It turns out that now an aquatic organisms actually have an increase in sensitivity, which is very worrisome. And so the group actually went and sequenced the target site, but they didn't get the entire gene. They just got some of the domains. And the important thing here is that strongly evidence for the impact of pesticide being transported into aquatic systems, and that there's some information about this particular gene vector. So if you pick one thing out of this slide is that you have an identified gene that I can go and find. So I queried NCBI in a general way, excuse me, to find out what do we know about this gene in general? What information is there available for the voltage-gated sodium channel? And I did go strictly to look for the genes that were available for arthropods, and then I found a few sequences. Some that came from the jewel wasp, Nasonia vitripennis. I unashamedly biased towards that because that was my organism of grad school. That's what I did. And then there's all the information that came from our infamous fruit wasp. In addition to information about that particular gene, there were some gene ontology terms. And so this gene, I know that it's obviously its molecular function is some channel activity for the voltage-gated sodium, and that it is involved in the biological processes of sodium ion transmembrane transport and olfactory behavior, as well as it is located in this that's just some more information that I got about it. From the supplementary material of the paper, I actually went and got part of the sequence. And so I retrieved the sequence of this part of the segment 3 of the second domain. They didn't get everything, but they got some sequences. It's a little structured, but about 94, 95 amino acids in this sequence, and so I'm able to just use that to query the genome. And to do that, I go to the window and I say search sequence, and this window pops up. The reason why I'm not doing this live is because I am cursed with the live demos, and so I'd rather show you the pictures instead of waiting there for everything to happen and talk and then internet to fail. So I've learned my lesson, so let's just do it on cooking show style. And so you use the search sequence tool to be able to input your protein sequence. Here you can do nucleotide sequence or amino acid sequence, and you will tell Apollo which one you prefer. And you can actually search all of the genomic sequences or just the one genomic sequence where you are located if you are already inside of one of the gaps. This is what that search result window looks like. You're going to see an area highlighted in yellow, and that area basically is letting you know where the extent of the significant sequence similarity is happening, and then you'll have a box. If it's a table that has representation of the results of the sequence similarity, if you've ever used BLAST, this will result very familiar to you. If not, it's just a table that gives you a score of the percentage of sequence similarity, as well as information about where that identity starts and ends in terms of coordinates. So you will go and click on each one of those results in order to be able to see what that similarity looks like. If you've never performed a sequence search similarity, then the one thing you will need to understand is that the score that gives you the significance for that sequence similarity will be better telling or more telling about the relationship if it is lower. So the lower the score, the better. Okay, and so I'll just click on one of those, and it actually will send me into the browser window and show me where the high scoring segment pairs are located. This is a customization. You won't actually see this today, but we can actually set this up so that you can have a track with all the results. What you will mostly see is this, where you can actually identify the gene of interest. So it is brightly highlighted in yellow. So you close the sequence search window, and then you can zoom out in order to see the entire gene model. So when you look at the actual browser and you display the gene models for that particular organism, in this case, we can see that those high scoring segment pairs actually overlap the portion of a gene. And so I accurately assume that that one gene is the one that is perhaps the one that is expressing the voltage-gated sodium channel. So I just select the entire gene and drag it up. You can drag it up, or the other way to bring an annotation up to the user created annotation area is the right click menu, and you just say create a new gene, and it'll put it up in the yellow area. This is the information that is available for this particular gene, but the different genomes will have different information. I'll tell you what you will be able to see for the Manakin genome. The Manakin genome will have plenty of transcription schemes, and then we'll also have alignments to other species as well. So of notice, we have the gene models that were created for this particular organism. This is called the half-speed or version blah, blah, blah models. You also have several alignments to different organisms, lots of things within the arthropod cluster and just outside as well. And I think there's Munoz-Torres there as well. And then we also have a little box that tells us that we've actually created a track that lets you see where gaps are located in the assembly, which comes very much in handy. And it also shows you the last 10 results. So we dragged our gene model, and we're looking now at the gene model. So the first thing we do is to retrieve the sequence so we see what is it that we got. So I get the sequence, and then what I obtain is a amino acid string. You can't read this from the back, but I'll tell you that these are the different types of sequence that you can obtain: the cDNA, the coding sequence, the genomic sequence that I was mentioning earlier. So you can just click through those radio buttons in order to change it, and then you copy that sequence from the top and put it on a public repository, on a public database, and query the database. My favorite one to query for everybody in the exercises will ask you to do this is to go to the NCBI blast site and query the entire non-redundant collection of proteins that are available from NCBI. When you do that, you will have three things. There will be a table of results that you're familiar with. It looks a lot like the one that I showed you earlier. Then you will have these two other things. And this is particularly important. It is called, it's an image of the conserved domain database. And this is important because it actually shows you what kind of information your gene already has. And so I'm looking at three little regions of iron transport, and there's the more sodium transport associated regions. And so it's looking like we recovered, at least we're getting some hints that we recovered the appropriate gene model. I am circling these at the end because when you actually look at the alignments, you can click on those red lines and look at the alignment. I'm comparing the query that I submitted from Las Becas with the crab, not very closely related, but sufficiently close to be able to have a significant alignment. And I can already identify that the beginning of my protein is located about 2/3 of the way towards the end of the subject protein, the one that is available. So keep that in mind. The beginning of my protein is already 2/3 of the way. The first thing that it makes me think is, well, I should probably go swimming upstream and trying to figure out if there's more information that I can use to recover the entire gene model, right? So it's not necessary that you read what's in here, but this is the gene model that we just looked at. Those are the last HSPs, or the blood HSPs in this case. And these are two gene models that are located upstream of the gene of interest that I just identified. So the most logical thing for me to do is, well, let's pick them up, figure out the sequence, conduct the same exercise, and let's get some blasts going on to see what they look like compared to the same sequences that I just queried. So I do the same, that one in the middle. So this is reverse thread, right? So switch your brain and start reading from your right to your left. The one in the middle, over here, seems to have an ion transport, so that's good news. This, I want to, because I know what that means, but it doesn't say in there, so I won't say until we look at it. And then the second one, or the first one in this case, all the way at the most 5' most end, also has an ion transport. When you look at this particular gene model, when I look at it, the first thing I identify is that the domain is actually broken. When you look at the domain, you see how this looks fragmented. Can you see that from the back, from the screen? It's actually jagged. It's not a round end. When you look at the conserved domain database information, that actually tells you that the domain has been broken. And so it has been on the bottom one. We're thinking left to right, right? So if we flip this, and we flip this, we actually realize that both ion transport domains have been jagged and they're incomplete on the same side. So it's starting to paint a picture in my mind. If I, in addition to that, look at the alignments in a very close up and personal way, this is the first one I showed you. My query is located about 2/3 of the way towards the end of that first gene model that I was investigating. And it's the same for that species, the crab, and also for the cockroach. Another lesson to take a look at. And then when I go to look at the very first, the one at the far 5' end, I also find out that the alignment of my protein starts to occur about 30 amino acids into the proteins that are available in the database. So for the crab, it's located about 35 amino acids into the sequence. For the mosquito, located about 65 amino acids into the sequence. So start putting that puzzle together. Lastly, that gene model in the middle, 6233. We can see that there are alignments as well. And it is actually located about halfway into the protein. How do I know what the protein looks like? Well, because every time I look at the results, you actually have the length of the protein that you just query, or that you have the significance of it. So we have three things. We have one gene model towards the beginning, one gene model towards the middle, and one gene model towards the end of that voltage-gated sodium channel. What do you think is going to happen? I won't keep you waiting. I know you're excited. There is going to, this is exactly what I think it's starting to look like, right? We have the first one that we looked into all the way to the end, middle one and third one. If we look closely at the alignments, halfway through, 2/3 of the way, and towards the beginning. It's starting to look like these three models need to be brought together. Yes? No? Is it clear? Have I lost you? Will you tell me if I've lost you?
What about those other exons? What do you do with those?
Oh, no. That doesn't mean that I'm getting just the first exon. It's just me highlighting that I'm looking specifically at the beginning, just to see whether or not it was just to show you an application of where the alignment was, where I was looking at it. But we're talking about the entire gene model, not just the exons, correct? That's good to clarify. So I have discovered then that because I have found significant sequence similarity to the same gene in 3 different parts and in 3 different locations of the gene models that I was looking at for this particular model in the database, then it's very likely that all of them will need to be brought together. To bring them together, I have to route one. I actually lift the gene model. This is the middle one. I can just double click on it to select the entire model, drag it into user-free annotation, and then bring the ghost towards the gene model that was already there. And then just let go and I will bring them together, or I select both of them, right-click and merge them. That's the 3rd time I've told you how to merge genes. I hope you now remember. The other thing that you need to do, is to figure out how the boundaries are going to be modified after you merge them. Okay? You brought them all together, but each of those genes actually had a start and an end. So this is when we go into the nitty-gritty. So this is me actually looking at every single exon boundary, realizing that there's one that has a non-canonical splice exon instead of DPAG. We're looking at TG. It should be GT coming out. We're talking from the reverse strand. And if I'm the intern, it's going to be DPAG. So that's wrong. And so what I do here immediately is to ask Apollo to automatically find the next donor. Again, if I'm an intern and I'm going from the reverse strand, so I'm going left to right, this is my donor and this is my acceptor. Those are 2 concepts that you're going to need to remember when you're looking for the canonical splice site. And so you just tell Apollo, all right, modify it and changes that splice donor in this case. What you see now is that, look at here how the gene model is green and highlighted. And now the gene model is light blue, just barely thicker than the one over here. This is difficult to see. You can stay close to that. So what has happened now is that the coding sequence has changed. There is no longer green, blue, or purple. What you have is this light blue. Like it. But then there's no coding sequence there, so you need to modify that as well. But you will modify that exon boundary. Then I also realized when I'm looking at the gene model that this particular tiny exon doesn't belong. We will not go into the particularities of how they are one base pair intron tiny little exon. We'll go with the more general assumption that the transcription machinery needs about 50 nucleotides in order to be able to engage in work. So by looking at this alignment and by looking at all the evidence down below, I figured that I need to take that exon as well. And then I actually decided that because it had removed the entire coding sequence, I decided that it was necessary to fix the translation start. So you just tell, I'm looking at the protein right click and say set the translation start. So from now on, all my edits will keep the actual coding sequence and only modify where it's located. And, oh, sorry, this is what it looks like after you set the translation start. So you now see that it's thicker. In purple, so different from that blue. And the other thing that I realized is when I'm looking at those alignments, as Morgan was saying earlier, towards the 5' end of the sequence, the similarity, the actual strength of the sequence similarity is going to lower. And so I actually looked very closely at those alignments and realized that there was a number of amino acids over here that were shifting the frame. And it was actually not letting me recover the entire sequence, so I moved them. And in order to move them, I just, again, tell Apollo to find the next downstream acceptor so that the protein can become a little shorter. recover more of the protein. That sounds very abstract, I know, but if you sit down and go through these steps with all the little guidelines and explanations, then you'll probably be able to understand it a bit better. This is then basically the summary of what we did. We had the entire gene model that had the HSPs, and we had the other two gene models, brought them together, and we conducted modifications in order to be able to solve the boundaries of each of the exons. I've already looked into the start and stop. I figure out whether the splice sites were kept, and then after I'm done, I just get the entire sequence and go back to the CDI and figure out, did I recover the entire protein or did I not? And then I add more information to the information editor, just all of the metadata that I was saying that it's important to capture. Remember whether or not you found other gene models, perhaps from the RefSeq collection that you want to add to cross-references. Did you read a paper about this particular gene model and this species, and you have now a pocket ID, put it in there. Did you find out that you actually have sufficient sequence similarity to let you know that you could associate a genotype term that assigns a putative function to this particular gene? Then you enter those. You'll comment whether or not you brought genes together, split them apart, enter a name and a symbol. We'll have to come to a consensus on how we're going to name our genes. For example, let's make sure that we don't add things like or similar to, right? And so we would like to actually create a standard for how we're going to name our gene models. And then in the case of some of the groups, we can actually ask somebody to review our gene model, or we can just say, I approve it instead. Okay, so here's how it looks when you're actually conducting this in the script. In the next minute and 45 seconds, you're going to see a video of how Apollo conducts an annotation, or how I conduct an annotation using Apollo. There's audio here, but it's likely that not everybody's going to be able to hear it, so there are closed captions down here, and what I'll do is read it for you as it goes, so that if you're in the back, you can see what's happening. So first we begin, First we begin by dragging the gene models that you're going to merge. So like I showed you before, you double click on them, and put them up in the user created annotation area. And that's how it looks like when you drag them. And then we know that these two predictions should be brought together to annotate the ortholog of RNA polymerase 2 in the honeybee genome. Then you click on each of the exons for those gene models, and then right-click to bring them together using the merge function. Merging the gene model recalculates the open read frame like I told you before, and the changes are here shown as orange circles with exclamation points in the middle. So those are the non-canonical splice sites that were just created. And then, the gene models are good examples for adjusting the splice sites using two different approaches. First, we're going to zoom into the base level using the right-click menu, zoom to base level. And you can select the boundary of this exon to the upstream splice donor. Just right-click on it. Then to set the upstream splice donor, then Apollo recalculates it. It is now a canonical splice site. You can go to the other exon. And we're going to use RNA-seq from nurse bees in order to figure out whether these reads are good enough to modify that boundary. So you click on the RNA-seq read, and you click on the exon, and then you ask them to set it as a 5' end. This is a reverse transcribed gene, and that's it. So now you've actually brought two gene models together, and then you've corrected the boundary, and you have same gene models. Is that easy or what? I actually created an exercise for you guys last night. I think it's like a few hours, but you'll run through it a lot easier because you haven't started. Okay, so that's it, at least from my end, in terms of how Apollo looks like. What are the biological principles you will be thinking about? What are the things that you definitely want to keep in mind when you're conducting the annotation? I wanted to, of course, thank the people that I work with, because by no stretch of the imagination would I ever pretend to say that I did all of this work by myself. I am the lead curator in a laboratory of software engineers, and Nathan Dunn is the technical lead. Susie's our boss. We'll have collaborators at the University of Missouri with Celtic Lab. We have a very crazy, fantastically talented developer at UC Berkeley. That is his professional picture that he gave me. And we have an incredibly smart collaborator at Washington State University who's working with a pilot or a framework project. This is just to pick a lot more people, but to highlight that this is all work that is supported by NIH and by the Office of Science of the Department of Energy of the United States Department of Energy, and a lot of people that I work with. So what we're going to do now, or when we're done with lunch, because we're 5 minutes away from breaking for lunch, is to work an exercise. So we won't start them right now, but I'll tell you something about it. Since we unfortunately don't have the Manakin genome, we're going to work with it. But fear not, even though we're going to work with it, this is exactly the same process that you're going to follow when you're annotating your team. As I said, I think when I introduced myself, I am species agnostic, because I do this with many, many, many different organisms. I just conducted this exact workshop with ecology project from the University of Sydney in the museum, and then before that I was with all of your best friends in Panama conducting this workshop at Strive for the Independence community, and for all of those projects we use a very well loved genome with incredibly good data set, and it just helps us to start as an example. We'll be able to conduct the same thought process and carry it out when we do the Manakin genomes. The only reason why I put the picture in here is to tell you that some of the sequences that we're going to be inspecting are bees, wasps, and to put that into a very, very violently grown and not very specific timeline, they're about 190 million years apart, and even then we still have sufficient sequence similarity to be able to use some of those sequences to pull out the sequence that I'm interested in from the wasps into the honeybee. So when we come back, I'll tell you more about the exercises. I'll send you an email over lunch, and you'll receive a PDF with set of exercises that are there. We'll have 2 instances to work with, and then unfortunately, this is far larger group that I am used to managing for the exercises, which means we'll probably need Farah. We'll have 2 different ones, and we'll have 7 exercises, but not everybody has to be working on the same thing. But it is possible that I will be working on my gene, and then shows up another gene because somebody else is working on the same region. As well as have collaborative tools that allows you to see in real time everything that somebody else is doing. And so that allows Chris and I to sit down and look and work on the same gene. I'll be in California and he'll be in North Carolina, but we're looking at the same region and edit it. And because of that nature, when we're conducting the exercises, you'll see each other's work. That's okay. I guarantee you that's okay. What we do is just label them. So when you get your gene of interest out of the user-created annotation area, just write down this morning work, or just call it when you want, or however you want to call it. But that way you can differentiate which teams the other people are working with. I'll divide people into groups. We'll have users and password information for us to get going. Okay, you're free. Go eat.
Yeah, let's come back at 12:30 instead of 1 o'clock.